Welcome back. So in this section, we're going to continue our formation of urine, go to the second step, which is known as tubular reabsorption. So what is happening in here, we are taking stuff from that filtrate we formed in step 1, and those substances move through the epithelial cells of the filtrate, going through those cells until they're in the peritubular tissue, from where they can then enter the peritubular capillaries. So if you recall, we have peritubular capillaries up there in the renal cortex surrounding the proximal convoluted tubule. So we're going to have substances being reabsorbed back into the bloodstream. That's why it's called reabsorption and not absorption. Now, in tubular reabsorption, the primary site is going to be the proximal convoluted tubule. As well, we are also going to involve the loop of Henle. And so, yes, the loop of Henle does tubular reabsorption, but in a physiology class, we like to talk about its specific process involved with tubular reabsorption, which is known as countercurrent multiplication. And we'll spend a lot of time going through that in detail. Now, we can have just a tiny bit of tubular reabsorption occurring in the distal convoluted tubule, but this would have to be linked with tubular secretion. So we're not considering the distal convoluted tubule as a site of tubular reabsorption, just the proximal convoluted tubule, as well as the loop of Henle. So starting first with the proximal convoluted tubule, this is where 99% of your amino acids, as well as your other organic nutrients, are going straight back into the bloodstream. We don't want them in the urine. You need them for food for your cells of your body. In addition, at least 2/3 or around 2/3 of the sodium potassium bicarbonate are going to leave the filtrate. And so when they leave the filtrate, they are going to go into the peritubular fluid to increase the concentration there before, through a process of diffusion, can they re-enter the capillaries. Now, glucose also re-enters the blood, but it's not quite as simple. Glucose that is up here at the top in the filtrate has to have a protein, this green dot carrier, which allows it to enter the peritubular cells. And then once it's in the cell, it then has to find a glucose transporter for it to exit the tubular cell to be in the peritubular fluid, from which it can then diffuse back into the bloodstream. So the fact that we have all this sodium, potassium bicarbonate, and glucose, steps 1, 2, and 3, all these things exiting from the filtrate through the tubular cells into the peritubular fluid, that is doing this osmotic pull of water from the filtrate. So now water is getting pulled out of the filtrate and will re-enter the peritubular capillary. And this is how, 60% plus of our water is going to be exiting the filtrate. So now we have a much smaller volume of filtrate that's leaving the proximal convoluted tubule. So here's how this happens very much so in the proximal convoluted tubule as opposed to the distal convoluted tubule. In the proximal convoluted tubule, this part isn't regulated. If we look back at our morphology of the cells in the proximal convoluted tubule, you will notice they have these tall microvilli, increase the surface area for absorption. Okay. And as well, there's lots of mitochondria because, as I said, some of this stuff requires active transport. It needs this energy source. And the tight junctions between the cells, well, they're not really quite as tight as we like to make them seem. In the proximal convoluted tubule, they're very leaky. So that water can have an osmotic pull towards that high osmotic number that's found in the peritubular tissues. And so this enables the diffusion. In contrast to this, in the distal convoluted tubule, we don't have very many microvilli, and the microvilli that are there are very short. In other words, we're not really significantly increasing the surface area for absorption or for diffusion. We're going to have to absorption or for secretion. We're going to have to use different mechanisms there. And as you notice, we do have mitochondria, but there's significantly less of them in the distal convoluted tubule. And the tight junctions here, they're actually tight. Water can't get between the cells. It would have to go into the cells. So what we do have in the distal convoluted tubule, which we'll talk about later, is we have receptors for various hormones that can affect the change. And the net effect of all these differences between the proximal and distal convoluted tubule is that water movement in the distal convoluted tubule is separated from solute movement, as in solutes moving, creating an osmotic pull works for the proximal convoluted tubule, but solute movement in the distal convoluted tubule does not create an osmotic pull where water can get through. So back to our tubular reabsorption. We're done with the proximal convoluted tubule, and now we are in the loop of Henle. Well, the net result is water and sodium is going to return back to the blood. But in order to understand this happens, I just need to hit a couple of points. Well, one big point. Between the descending limb of the loop of Henle and the ascending limb of the loop of Henle, there's no blood vessels separating stuff. There's no other cells of the kidneys separating them. We only have peritubular fluid separating them. So what is happening concentration-wise in those peritubular fluid can directly affect what's happening inside those tubule. And so the exchange between these two, as in the descending limb of the loop of Henle and the ascending limb, this exchange is called the countercurrent multiplication system, which sounds really complicated, and it sort of is, but really isn't. Okay, so what you have to think of is we're going to have an exchange of fluid that's moving in opposite directions. And the descending limb is moving down towards the medulla of the kidney, and the ascending limb is moving back up towards the renal cortex. And so if you go back and you think about the basic anatomy, we had two different types of nephrons. We had our cortical nephrons, which had really short loops, which only went a short distance. And then we had those juxtamedullary nephrons, which had those really long loops of Henle, which went way down towards the renal pelvis. And so this countercurrent multiplication occurs because the juxtamedullary nephron loops are going to create a concentration gradient in that renal medulla. And this multiplication basically is an effect of exchange that increases as the movement of fluid increases. So the movement is going to happen in the nephron loop. That's the countercurrent multiplication. But now if you recall, that these nephron loops, as in the nephron loops, the really long ones of the juxtamedullary nephrons, they are surrounded by a second capillary mechanism known as the vasa recta. And these vasa recta are going to preserve that exact gradient through the depth of the renal medulla. And it's this gradient that allows for the formation of the highly concentrated urine. So there's actually three key players going on in here: the long loops, the vasa recta, and the fact that the collecting duct can use this gradient to adjust the urine concentration. So let's look at this in more detail. And in order to understand this, the key is don't start where it's descending, but start with the last part of the ascending limb, that thick ascending limb, where we have those cuboidal cells. And in the thick ascending limb, we have a lot of active transport going on. So what we have is we are using energy to move sodium out of the filtrate and into the cells, as well as chloride. And then once they're in the cells, we're going to use active transport one more time to secrete them out the basal portion of those tubular cells. So now they're in the peritubular fluid and they're in the renal medulla. And this is very effective. And in fact, almost 2/3 of the sodium and chloride can be removed. 2/3 of what remained when it entered this area will be removed before it reaches the distal convoluted tubule. So remember, we already lost 2/3. So of the 1/3 that's remaining, we're going to be removing an additional 2/3 of this. Now, in addition to the active transport being required and moving tons of sodium, potassium, we need to realize that this apical surface in the thick ascending limb is totally impermeable to water, as in all the water that is still there in the filtrate will stay in the filtrate. There's no water entering the cells, into the peritubular fluid, going into the blood. There's no osmotic pull whatsoever. Water cannot leave the filtrate in the thick ascending limb. Okay, with that, we're done with this part. If you understand this, then we have a good basis from which to understand this countercurrent multiplication. So, because water can't leave, but all the sodium and potassium has left, this now means that the peritubular fluid is very high in solutes. It has this high osmotic concentration going on. So if we go back and we look now at the thin parts of the descending limb, what you will notice in this picture, if you compare the descending and ascending limb, you will notice that the thick part of the ascending limb starts quite low, whereas that thick part of the descending limb ends quite high in the renal medulla. So that there is a lot of thin descending limb in the parts of the renal medulla where we have that osmotic movement. We have all those molecules of sodium and potassium in the peritubular fluid. And so we say we have this increased osmotic concentration in the peritubular fluid, which is surrounding the thin descending limb. And this is showing gradients in the peritubular fluid. And so what is going to happen is this thin descending limb is permeable to water. So water can enter, water can leave, but solutes cannot enter. So all that sodium, chloride, potassium, whatever might be in that peritubular fluid cannot enter the thin descending limb. And so as the tubular fluid travels deeper and deeper in the descending limb, in that thin descending limb, it's going to have more and more osmosis into the peritubular fluid. In other words, because we are surrounding the thin limb with all this sodium and chloride, we are going to be losing more and more water as it dives deeper, thereby concentrating the fluid inside the descending limb. And so on this one, you can see the numbers changing from being very low to being super concentrated by the time it's hitting the hairpin curve of the loop of Henle. And then it will start ascending in the thin ascending portion, but as soon as it hits that thick portion, now we're going to have the active transport of sodium and chloride back out of it. So to review this countercurrent multiplier, you have to recall that sodium is constantly being added to the proximal convoluted tubule by the glomerular filtration. And then the higher the concentration we have here in the peritubular fluid, the more water we will have exiting the thin descending limb, thereby concentrating the fluid that remains in the tubule. So we have a super salty filtrate entering the thick ascending limb, thereby loss of active transport to pump it into the extracellular fluid. And the more salt that is pumped out of it, then the saltier the concentration or the more milliosmoles we have in the extracellular or the peritubular fluid in the renal medulla. Okay, so pause it if you need to for a second to make sure you really understand this before we add the next step. So in the next step, we are talking about the vasa recta. And the vasa recta basically are just an exchange mechanism by which we can maintain this. So if you notice, and I need to put my glasses on here so I can see the what I am doing. If you notice in the descending thin limb, we are losing a lot of water into the peritubular fluid. And then in the ascending limb, we are losing a lot of sodium. So proximally, we have high sodium going here. And so sodium can go into the arterial and water can exit the arterial in the capillary. On the arterial side of the vasa recta capillary, what we are going to have is we're going to have water exiting and sodium entering to keep the concentrations the same. And then when we get up to the venous side, we are going to be losing that sodium that we brought in and reabsorbing that water. So the fact that we are reabsorbing the water at this level where we were releasing the water means the net water movement in the peritubular fluid is the concentration remains the same. So we are just exchanging things to keep gradients. So with that, I'm done with the second step, and I will see you shortly with the third step, which is a lot less complicated. So thank you for all your hard work, especially on this second step.